now I am pleased to introduce Michelle Geary. Michelle is a plant ecologist who did her doctoral research on tan oaks in Mendocino County and currently leads research teams studying coast redwood seedling survival and mechanisms for how plants deal with excess light stress. Since 2003, she has been teaching plant biology, environmental biology, and cellular molecular biology at West Valley College in Saratoga, California, in the shadow of the Santa Cruz Mountains. And we are also happy to be joined today by Hugh Leonard. Hugh started at West Valley College and transferred to UC Santa Cruz and received his BS in biology in the spring of 2020. Hugh is starting his doctoral work this fall at UC Santa Cruz, looking at how mechanisms for dealing with water stress affect survival of different tree species in California forest ecosystems. He has been one of the team leads for the West Valley Redwood research team since early 2018. And with that, I will pass it over to Michelle and Hugh.
So obviously, I'm Michelle. And what I'm going to be doing in this talk, it's not just a science talk. I'm going to be talking a little bit about who we are as a team and what we're interested in specifically. So we are the West Valley Redwood team, and we're studying coast redwood seedling response to climate change. As I said, we're going to start talking about not science specifically, but studying science. We're going to talk about undergraduate research at West Valley. Then I'm going to give a big picture overview. I'm going to put the project that we're doing in its overall context, including talking a little bit about why we do what we do specifically. And then Hugh is going to talk about what we've been doing very specifically at Pepperwood and some of our preliminary results. So can you all hear me okay? Okay. So let's talk about undergraduate research. So when I'm talking about undergraduate research, I'm focusing on STEM, so science, technology, engineering, and math. And when you talk to people about what STEM students do, oh, this person's a chemistry major, this person's a biology major, people are thinking they're taking really hard classes, which well may be true, but taking a hard class and doing well in a hard class is not necessarily science as such. And so there's been a movement over the last, I want to say, decade, saying how can we enhance the research experience of undergraduates? How can we give them a chance to actually do real science so that they are better prepared for their future careers? And so one question that I think naturally comes to mind is when I'm saying science, what do I mean by science? So asking and trying to answer questions by going through an iterative process, trying one thing, running it again, trying it again, fixing it, having it be hands-on. Real, authentic science is going to have problems. You're going to try this, you're going to discover, oh wow, I wish we had done such and such down back at the beginning. Real authentic science involves a lot of failure. And if we want to train scientists, if we want to focus on enhancing critical thinking, as opposed to doing a cookbook lab, we need to be giving students the opportunity to do real science. Now, I want to, at the very end of my talk, there will be a slide that talks about some of my team leaders. The young woman that is crouching down, looking at that thing, which is called a rad shield, and I'll explain that in a second. This is Joycelyn Ho, who is one of our long-term team members. Nick Bergeron is also in that photo. And down on the lower right is John Adams, who is our seedling team lead. What's happening in the photo with John is that we're building things, building rad shields, which protect our data loggers from direct and indirect sunlight so that they can measure things precisely. And so we cut and drilled and sawed and made mistakes and worked on it again. So constructing, testing, what Joycelyn and Nick and Hugh, who was cropped out of the picture, were doing in this photo, we were testing the rad shields to see whether or not they were in fact protecting the data loggers from direct sunlight. So real science involves failure. Real science involves not necessarily knowing what's going to happen and plowing forward and looking at the results and analyzing the results and then running it again, that iteration, to see whether or not, just to see what's happening, to answer those questions. So we need undergraduate research in general. And especially, obviously, I'm going to be focusing on that for STEM students. So how do undergraduates get research experience? So if you go to UC Berkeley or UCSC or Davis or Stanford, there are a lot of professors that are engaged in research. So they have research labs, and many of them offer chances to work in those research labs to begin getting research experience. Many colleges and universities have research educational opportunities for undergrads, REUs and REUs, usually funded by NSF or the USDA. And these programs can give students a stipend, they can give experience. They're often very competitive to get into, which means that students are going to be more likely to get into these if they've done work in research labs. There might be outside internships with nonprofits, with groups like Pepperwood, with biotech labs. Those outside internships are also going to be interested in students who have experience. So those first two things, the internships in research labs and those summer programs, are often things that are seen at four-year colleges. And the first three, obviously, are going to be easier to come by at a four-year college. So there are a lot of ways that undergrads can get research experience. The last one that's mentioned there is having research actually embedded into the coursework, embedded into the semester, so that students can understand that process of doing things and not knowing what's going to happen. My husband asked once, I was telling him about an experiment that my class was doing, and I mentioned as part of this discussion that I didn't know what was going to happen. And he looked at me like horrified. And he said, "What do you mean you don't know what's going to happen?" I said, "It's an experiment. I don't know what's going to happen." And he said, and I quote, "They let you do that?" I guess they being the administration or whatever. And so I thought, on the one hand, number one, I'm not sure that they know. But number two, this is a significant movement, giving students a chance to do at least a little bit of research. Those little tiny plants over there on the right, by the way, these are, I'll mention them and again in just a minute. These are Arabidopsis. They're a model organism. And I have my students work with Arabidopsis in my plant biology course. I've also helped run an REUU funded by the USDA that is theoretically housed in South Carolina that works on Arabidopsis research. So undergrads can get research experience in a lot of different ways, but it's harder to do if you're at a community college. So research at colleges is not common. And I'm going to say that it's really important. One of the reasons it's not common is that faculty do not have their own research labs. We have a natural history lab that has a green room attached, and I pretend that that's my lab, but it's not my lab in the same way. Faculty are being paid at a community college to teach full-time. They're not being paid with the idea being that part of their time is being devoted to research. So although my college supports what I do, it's in a sense that I've done. So research at community colleges is not common. One of the reasons that it is important, as well as nurturing young scientists, is that it helps us tackle some of the issues with equity. Students that are coming from traditionally underrepresented groups, students who are the first in their family to go to college, are more likely to start at community college than at a four-year school. What this means is, traditionally, they would not have research opportunities. So Our students, if they're transferring to a UC, they usually transfer after 2 years. If they're transferring after 2 years, coming in as a junior, they will not have had true science experience. They may know techniques. If they're applying for an internship in a lab, they are generally less competitive. This is something that I've done, I don't know, post-grad plus 2 years type interviews with former students. What happened when you were there? What did you notice? What should we be doing more of? So after they transfer, they're less likely to get internships in research labs. So now they're less competitive overall in terms of getting into grad school and in terms of getting different kinds of jobs. So from the get-go, if they don't get the experiences, they're behind. And they're already behind because they're coming from these underrepresented groups, and they don't have the kind of the unspoken curriculum that students that are coming from families that have gone to college forever perhaps have. The other reason why research opportunities are important is that what data shows is that if you have what's called open-ended research, stuff where nobody knows what's going to happen, where no one's coming in having done that lab before. If you have open-ended, authentic research experiences, it increases retention of students from underrepresented groups. They stay with it, they persist, they don't feel like they don't know and everybody else does. It increases success, meaning that they are more likely to pass the class. And again, it's going to make them more competitive when they transfer. And this is what we need in STEM, is to be creating pathways for students coming from these groups to succeed. So it's unusual. How did we get into this? So when I started teaching at West Valley, it was, I'm going to say, sort of an accident. I fell in love with the place. I knew that by being there, I was basically setting my life up to be primarily teaching and not research. So I wanted the chance to do research again, and I wanted a chance for my students to have research opportunities. I started doing a little bit of here stuff and a little bit of stuff outside the classroom. I have what I call the Little Plants Project and the Big Plants Project. Arabidopsis is the little plants. In the Arabidopsis group, we're looking at different genetic mutants and looking at their ability to do photosynthesis under different kinds of environmental stresses. As I began working on both the little plant group and the big plant group, the Redwoods, I was trying to think about different questions that we might be able to ask and try to answer. And I began recruiting in my classes, saying, hey, I'm doing research, you could do this too. Now, the big plants, you will not be surprised to hear, are of course the Redwoods. And I was interested in Redwoods in a general way as a Californian. The young woman on the right-hand side is Jennifer Moe, and she was the first person on the Redwood Project with me. And we were interested in what was going to happen with redwoods and climate change. So, okay, let's do some research. We want to do research that means something. It's going to answer important questions, but we don't have a lot of money. We don't have a lot of fancy equipment. It can't be too far away. We can't go to Puerto Rico to do field work, for example. And we don't have graduate students whose experience we can draw on. We don't have juniors and seniors who have already taken all the biology classes. So we're going to be working with students in this project that don't have a lot of experience. And as we were going through this process of what are we going to do, one of the things that Jen and I realized is that seedlings are easy in the sense that they're small, they're on the ground. You don't need to climb a tree. You don't need to have fancy equipment to count or to measure height. So the project started, the projects have built. You'll see a lot of names at the very end. As I was doing this, I was thinking, how do I make this work? How do I deal with the fact that students are with me for 2 years and then they usually will leave? So I thought of, students that were first year, students that were second year, students from the second year, training the students from the first year. So I was thinking of this entirely in a practical mode. I will have multiple people and I will have them train each other. This is important for their learning. But things happened that I hadn't expected. The team became a community. We have learned together, we support each other. It has become something that for a lot of us, and notice I'm including myself there, it's emotionally important. We think that we believe that we are doing meaningful work, that we're going to help solve significant problems. This has been valuable for me personally in the sense that I have reestablished old connections. I have felt like I'm, I don't know, reaching my full potential. So if there's a personal growth aspect to this, the picture over there on the right is John, who we saw in an earlier photo, Hugh, who's going to be talking to you, and Amaya, who's one of our newer members on the team. And we went to Sequoia Kings Canyon, and we were setting up long-term monitoring plots to look at seedling survival of giant sequoias there. So for me, this has been something that has been an amazing experience. So research is educational, research is valuable, and research is fun. Maybe you're out there getting grubby, getting tired, hiking for too long, getting ticks and the whole bit, but research is a fun experience. So I am now going to shift gears. So we're going to talk about the broad context. Now, this is a photo that that John Adams took. We started this spring a collaboration with the California Native Plant Society, setting up long-term monitoring plots in the Santa Cruz Mountains in post-burn sites to follow what happens in those areas after fire. And so we'll be talking about pepperwood for results primarily, but this is something that we are doing in multiple places. If you know how big redwood needles are, you can see that there are some in that kind of fuzzy forest floor down there. So this seedling is like this big. It's incredibly small. So why does the future of coast redwoods matter? Now, we're Californians in the sense this goes without saying, but they have iconic status. They are part of what we think of when we say California to people around the world. They think, oh, you've got the really big trees. We have lost a lot of redwoods. We have, I think, less than 10% of the old growth that was available in the past, that was present in the past and before European contact, is still there. And much of the range, there's the potential for restoration. I think somebody mentioned that one of the reasons he was here is that he's hoping to plant redwoods on the property. If we understand what redwood seedlings need and what redwood seedlings need as the climate changes, we can do restoration that has a higher chance of success. We can do planting and restoration that is going to be more likely to help us achieve our goals. As an ecologist, one of the goals that I'm interested in is the fact that redwoods store carbon. So planting, helping create, helping restore more healthy redwood forests can be one of the ways that we tackle pulling carbon dioxide out of the atmosphere. So they're iconic. We've lost a lot, but we can restore. We can store carbon because we know that redwoods are one of the absolute best ways to store carbon. There was research that came out in the last year indicating that. But if we're going to have these redwood forests doing all of these important things, we need to know what's going to happen with coast redwoods as the climate changes. So here we have a map, and that lovely aqua color is more or less the current range of coast redwoods. And so you can see that there's data coming from a number of sources mentioned down there in the caption on the bottom. Many of you will know, and it is probably kind of obvious from the photo, that redwoods, the coast redwoods, are restricted to the range within which fog can percolate in, or at least could percolate in historically. Those different locations, I mean, if you go to Mendocino County versus the Santa Cruz Mountains versus the areas along, down by the Ventana Wilderness, those different locations have different temperature environments. They have different rainfall patterns already. So one of the questions with what happens to redwoods is what's going to happen as it gets warmer, as temperatures increase? What's going to happen as precipitation patterns change? One of the things that we're seeing is that the first rainfall of the fall is happening later and later over time. And one of the big elephants in the room is what's going to happen if the fog pattern changes? And I'm going to tell you right now that, and I will tell you again in a second, that we don't know a lot about what the fog pattern is going to do. So notice this slide here does not have any pretty pictures. This is a grim slide. This is a, let's think about this for a moment slide. So the maps on the next page are based on research that came out in 2017. And what they wanted to know was, if we imagine redwood seedlings out there in location X and location Y and location Z, which areas will be suitable places for redwoods to survive as the climate changes? Until recently, when I was using the climate change models, I was using climate change estimates based on a low emissions scenario, the idea being that we would have dramatically cut our carbon emissions, what's also often called the B1 scenario. That is not the trajectory we're on. So the estimates that I'm going to show you are estimates based on the high emissions scenario, which is the trajectory we are currently on. So which habitat would be best for, suitable for redwoods to establish as seedlings in the open under climate change? There are flaws, and I put flaws in quotation marks because the researchers knew that there were limitations. There were things that were not addressed in their research. They do not take into account in their models the effect of fog because there's so much uncertainty about what's going to happen with fog. We've seen reductions in fog over the last 70 years, but some places seem to be getting foggier. And depending on what happens with ocean currents and upwelling, we might actually see increases in fog at some parts of the year at least. The other thing is that redwoods as individuals are incredibly resilient. They live for a long time. They can come back after fire. And so it's not just about, can seedlings establish? It's also about, can the adults hang on? And if the adults are hanging on, they're changing the microclimate underneath the canopy. And they're creating refuges, there I call them micro-refugia, where seedlings can potentially establish. So I'm going to show you in a moment the incredibly depressing maps for suitable habitat for coast redwoods to establish. And it's scary. And it doesn't reflect what I think is going to happen, except in the worst case scenarios. Okay, so I'm going to show you the maps. You need to remember redwoods are crazy. This is a redwood that was very badly burned, and notice it is sprouting new leaves and new branches all over, even though it was burned to the very top of the canopy. Okay, let's look at a map. So we have three maps here. The first map, the one on the far left, is that same map that I showed you before, the current range. What I did in the other two maps is I, to a different degree, I emphasized the outline of the current range so you could see it more prominently. In that center map, we have the current emissions scenario. If we stay on the trajectory we're on, what's it going to be like in roughly 30 years in terms of suitable habitat? And those yellow blocks are the areas that would be suitable habitat. Notice that there's an awful lot of the Redwood Range, including Sonoma County, that no longer be suitable for redwoods to be able to be easily established. So the 2050, we're losing area. The climate is changing. It would be harder to do restoration in those areas. It would be harder to get those forests thriving. That map over there on the far right That's the 2090 scenario. Now, the way that they entered the data for this particular model creates these little blocks. But basically what it's saying is the only areas in California that would be suitable habitat for redwoods to straight up establish is where the red is. Like I said, they're not taking into account fog. They're not taking into account that the adults have the potential to create conditions that seedlings can survive underneath in these refuges. But still, this is the direction that we're currently headed in, that habitat will become less and less suitable for redwoods to be surviving in. So for our forests to hang on, what do you need to have? So you need to have the adult trees surviving. And I've got ordinary in quotations. This basically means seasonal conditions. Can they survive how cold it gets in winter? Can they survive how hot it gets during the summer? Can they survive what's happening with the rainfall patterns and the change in the rainfall patterns and the change in the humidity levels? And what they're going to need fog in many places. What will happen with the fog is a big question. So for a healthy forest, we need the adult trees to be able to survive. Some of you may have noticed that a lot of the redwoods, say along Highway 101, are looking really stressed. We're in year 2 of a drought, and we only had a couple years of more or less normal rainfall after the last drought. Maybe what we're seeing is that we're at the limits for what some of these adult trees can really survive, but we don't know. The other thing that has to happen to have our healthy, resilient forest is we need to have the adult redwoods coming back after fire, the way that you see in this photo over here on the right. Redwoods are also really good at coming back after flooding, but maybe the floods with changed rainfall patterns, maybe those floods will be faster, maybe they'll be different, maybe they'll occur more frequently. So in order for our forests to be able to survive long-term, we need to know that they can come back after disturbance and come back after disturbance in different parts of the range. You also need to have juveniles showing up. Maybe these are sprouts from damaged stumps. Maybe this is seedlings, because if you're going to have long-term population survival, and I'm thinking about thousands of years here, you need to have seedlings. And this, of course, is where my team is focused. So for long persistence, you need improvement, you need new individuals. Those seedlings are going to give you genetic diversity. For seedlings to survive, there are a couple of different critical stages. The trees need to make seeds. Not all seeds are going to be good quality seeds. There's data suggesting that under really hot conditions, seed production may decrease. You need to have the seeds germinate. Data suggesting that under high temperatures, the seed quality may be poor, and the seeds may not actually begin to grow. Assuming you have a viable seed, assuming that it starts to grow, what determines whether or not it's going to survive? Over there on the right, on that tape measure, that's in metric, by the way, so these are centimeters between 39 and 40 and 41. That is a redwood seed that is just having a tiny seedling emerge. These are very tiny seeds. They need to fall in the right location, usually on comparatively bare soil. They need to start to grow. And for long-term survival, you need to have what's called establishment. They need to be surviving. Hugh's going to talk about this. They need to survive the hot summer. They need to survive the cold winter. They need to survive the drought period. For a lot of people that study conifers, there's this basic question: how long before you can kind of say they're good, that those individuals are going to persist? And I'm going to say that around 3 years is probably good. 2 years maybe, and Hugh's going to touch on that in his talk. So not all of the coast redwood populations are really at risk. right now, in the northern part of the state, we're actually seeing an increase in how fast the trees, the adult trees are growing. It's cold up there. And with warming, they're seeing more time, more of their days are at temperatures that are good for growth. There's more carbon dioxide in the atmosphere. So they are doing more photosynthesis. And in those locations, they're seeing some seedlings, but usually only in really high sunlight areas. They're getting fog, they're not too hot. Those seedlings, I'm glad that they're doing well, but they're not really interesting to us in terms of the questions we're asking. We're interested in the fringe. This map I'm using because you can look at that brown versus the green edge, that edge on the east, those edges on the south. Those areas, those fringes are going to be hotter, they're going to be drier. We can see what seedlings are doing in those locations and get kind of a snapshot of what might happen in the future, as well as what actually is and is not happening right now. And that's why we're interested in the locations we're interested in. So currently we have 3 main areas we've been focusing our work. In the south, we have a site near Big Sur at a UC reserve. In the area near where we are, we have our original site at Bear Creek Redwoods, and then we've been moving, as I mentioned, into the Santa Cruz Mountains and looking at post-burn recovery. And of course, we have work that we're doing at Pepperwood Preserve, and Hugh's going to be talking about that in his talk. So I've got senior team, Jen that started this with me, Hugh, you're going to hear from, John, Joycelyn, Brandon over there on the right. Joycelyn, I don't know if I mentioned this, is actually a marine biologist, but she comes and plays with us. Brandon is at UCSC right now, and he is a herpetologist, but he hangs out with us and does work too. Over on the left, we have the people that are currently more active on, either they're at West Valley or they're coming to things with us within the last, say, 5 months or so. We have new team members that have been recruited. New in this context means we have not dragged them out into the field yet, but they've come to training or been, they're on the mailing list. And then we have people that have been involved with the project in the past. So I started out with 1 person and the project blossomed. Thank you so much to West Valley College for supporting me philosophically and sometimes with equipment, to Pepperwood, of course, to Mid-Penn Open Space District for letting us use their site, to Landals Hill, to the California Native Plant Society. And for some of our funding, we did fundraising at a site called experiment.com, and we appreciate everything that they have done for us. So I've talked about the undergrad research, I've talked about the context of the project, and I am done.
Thank you, Michelle. That was wonderful. And I will go ahead and start my presentation now, and I'll be focusing on the specifics of Pepperwood as it is. And I will clarify now that my video is on now, so you can see a face. I can associate that with my voice, but I'm actually going to turn it off during the presentation due to rural internet bandwidth issues. Do that and then start this presentation. Okay, so as I said, this is an outline of what I want to be talking about, specifically focusing at Pepperwood for our research so far. And this is a bit of an outline for how I want to go through this talk, which will be to present our experimental design and how it has changed over time, show some interesting results and findings that we've had over the years that we have been out at Pepperwood, and then discuss some future avenues for research going forward with our research there in the context of our findings. So first off, why Pepperwood? And Dr. Geary has already mentioned this, but we wanted to look at Pepperwood specifically due to its location on that eastern boundary of the current redwood population range. Pepperwood occupies one of those fringe habitat sites and is interesting in a habitat sense to look at the context of microrefugia, future climate change, and those model predictions for a reduced redwood population range. So by looking at Pepperwood's microclimate conditions, we are seeking to gain that insight into what the future climate conditions could be for those non-fringe regions that will be facing a warming and drying climate. Now I want to clarify that term microclimate. Typically when we hear that term, it tends to be more like the microclimates of a region, say the San Francisco Bay microclimates. What we're talking about here is at a more granular level, because we're interested in knowing what are the temperature, the relative humidity, and canopy closure that are being experienced by seedlings at a specific forest at a given point in time. We're also interested in knowing how disturbances can affect these microclimate conditions. And one of the most major disturbances that we are all very well aware of here are fire. So in 2017, of course, Pepperwood experienced a dramatic change from the 2017 Tubbs fire. However, this also presented us with an opportunity not only to investigate the fringe population of redwoods, but also to investigate the microclimate conditions that could have been altered due to this fire. And interestingly enough, the Tubbs fire didn't just affect Pepperwood, but it affected it in a homogeneous pattern. Part of the redwood population experienced a high burn event or high intensity burn event that drastically affected the forest structure. And in this picture, you can see that the entire canopy was actually removed at this site. However, a portion of the redwood population received a low intensity burn event where the canopy remained intact. The high intensity was at the Grouse Hill region of the preserve, and the low intensity was at the Redwood Canyon region. And I'll use those terms, Grouse Hill and Redwood Canyon, because that is typically how we associate our research when we talk about it with our project. So now we have not only the opportunity to look at how fire also affects these fringe microclimate conditions, but also how fire intensity can affect seedling recruitment at these different microclimate sites and this same geographic region, which is a very unique opportunity to pursue. So to do this, to capture this microclimate variation, we deployed what Dr. Geary talked about earlier in her presentation with these data loggers. Now, this is not the data logger itself. These are the radiation shields having been deployed in the field. The data log itself is just a tiny egg-shaped device used to capture relative humidity and temperature on an hourly basis inside these sites. We deployed a series of data loggers at both our Grouse Hill and our Redwood Canyon site, and we deployed them along an elevational gradient to help capture if there might not just be variations in a specific microclimate region, but also to show us what kind of variation in microclimate data occurred at the site. We also took canopy closure measurements, which is using a device called a densiometer. And what this effectively allowed us to do was know how much light was penetrating to the forest floor through the existing forest structure at both Redwood Canyon and our more intensely burned Grouse Hill site. We then deployed these also at the beginning of the summer dry period, actually late spring, to be able to capture that most extreme event in terms of seedling recruitment and establishment at these microclimate sites and for this region. And then finally, we did observational surveys to see what, if any, seedlings had become established or had been recruited, rather, at both Grouse Hill and Redwood Canyon. What we gained from this 2018 data collection year showed us two interesting aspects of the preserve. First was, taken from our data logger data, was that the amount of variability in both temperature and humidity was far greater at our Grouse Hill site, which was the high intensity burn, compared to the low intensity to the moderate intensity burn at Redwood Canyon. We had actually expected this to be the case due to that complete removal of the canopy and that alteration of the forest structure to such an extent that Grouse Hill habitat was no longer able to provide any climatic buffering for variations in extremes for humidity or temperature. We also conducted from those seedling experiments, we observed rather that there were no seedlings being recruited at Grouse Hill, largely due to that fact that it was such a high intensity that all the existing cones and seeds that appeared to be present in the seed bank were consumed or no longer viable. The alternative site at Redwood Canyon showed the opposite result. We had many new seedlings maturing or being recruited at that site early in the season and throughout the summer. But it also had experienced a high mortality event later in that same year, where a majority of the seedlings did not survive, but a select few did. And what you can see in this lower right-hand picture is a tiny little seed with its tiny little germination coming out and just poking above the soil. So this was really great for us because now our goal was really to go out and look at this plant microrefugia, see if it's supporting seedlings. But now we also had more questions, of course, as to what were the conditions that were allowing some seedlings to survive, but also leading to this high mortality event. So in 2019, we augmented our research design to further investigate our findings at that Redwood Canyon site. We established a dedicated 20-meter square plot inside the existing redwood forest envelope, and you can see that in the top right picture. And then within this dedicated 20 by 20 meter site, we created two 3-meter square seedling monitoring subplots, which you can see in the lower left is when we were just beginning to establish this, or lower right, my apologies. These were opportunistically selected so that we could capture seedlings that we had observed growing early in the spring season, but they were also opportunistically positioned so that we could capture an additional aspect for seedlings, which was light availability. One of our subplots would be oriented next to and within the existing redwood canopy, which was much more dense, and one was located just outside those adult redwoods, but received more diffuse light. We then took measurements of each of the seedlings inside that plot, or each subplot, and those were height measurements to assess above ground biomass accumulated at that point of initial installation. We geotagged each seedling for our future reference, and then in subsequent trips would come out and remeasure the height gain for each of the seedlings, as well as the mortality of these seedlings over time, so we could specify when it occurred for each seedling. So at the end of our 2019 data collection year, our results showed us some very interesting trends as it relates to humidity. As in 2018, 2019 had also experienced a high mortality event with a subsequent high recruitment event earlier in the year. But what was interesting is that the data suggested that the most mortality occurred at the lowest relative humidity measurement. We recorded a measurement of approximately 12% relative humidity. And you can see that in the upper right graph, where the blue line represents June through July for relative humidity per day. and the orange line represents September through October. And that lowest point in early October was the point where we see also a corresponding drop in our seedling survivorship, which you can see in the bottom right. Now, I mentioned here that also occurred at the closest approach of the Kincaid fire to our research plot. And it might be assumed that the fire could have affected this, and very well might have affected that really severe drop in relative humidity. However, I do want to stress that in 2020, we had a similar decrease in relative humidity into the mid-teen level. And it appears that fire was not a corresponding factor that necessarily to this low humidity drop. So this is the end of our 2019. So we know that, or think that, humidity plays a factor. So if only there was a location that we knew had a lot of variability in humidity. Well, of course, there is. So we refocused in 2020 at our location at Grouse Hill due to that high variability in both temperature and humidity relative to our Redwood Canyon site. Now, we knew that there were no new seedlings being recruited there due to the consumption by the fire of all existing seeds in either the consumption or inviability of seeds that had survived at Grouse Hill. But what we were curious to know was, is this habitat still capable if there had been seeds of new seedling recruitment and establishment? Or in other words, had the site become so altered that at this point it was no longer viable for new seedling support? So to answer this question, we devised a seedling transplant experiment for our Grouse Hill high severity site. We sought for and were approved to do a transplant of 24 seedlings into 3 separate plots. And these seedlings had been cultivated, raised to the 1-year mark, and then were transported for transplanting. Additionally, not only were we going to conduct this seedling transplant experiment, but we had recently read newly published research indicating that soil microbial communities and the microbiome can be very important for new seedling recruitment and establishment. But also that high intensity fire events can damage the soil microbiome community. So to address that factor as a potential confounding variable, we actually created a paired trial for each seedling with another seedling that had been inoculated with soil taken from our Redwood Canyon site, where we knew seedling recruitment and establishment was successful. So for each 3 of our plots, there would be 4 control seedlings and 4 inoculated seedlings. In addition, we also placed these new plots opportunistically to take advantage of different potential microclimate states. One would be positioned inside the existing redwood envelope. One would be positioned adjacent to, but outside of that existing redwood envelope. And one would be placed in a completely alternative site that was not associated with redwoods. We then took initial height measurements to record above ground biomass, and then geotagged them and placed a data logger within each one of these plots to be able to monitor those microclimate conditions over time. We did not make any other alterations to these plots, and that will be important for what I'll talk about in our results. And then we allowed this experiment to run for the remainder of the year and assessed at the end of our 2020 season. Unfortunately, what occurred, or fortunately, it's science, it's not really directional, is that none of our seedlings, regardless of location or inoculation treatment, survived to the next year. I'll also note that we planted these in February of 2020. so that they could take advantage of any residual soil moisture without being shocked and then trying to survive through the dry period. However, that was still not enough for these seedlings to have survived to the next year. Initial observations had indicated that the inoculated seedlings had seemed to fare better in terms of color and resistance to the drier periods, but it was not as significant a result to be able to extend to survivorship. So what our conclusions from this trial so far have been that the factors of either light intensity and or water availability and or some other yet determined factor are more important at our Grouse Hill site than at our Redwood Canyon site for seedling establishment. So now, notice that I said that this was in 2020. 2020 was a rather difficult year, I'm sure, for all of us. And the reason I talked about making no additional alterations to this site was because we weren't actually able to necessarily make any other adjustments to our experimental program before March of 2020 rolled around, and then COVID-19 pandemic madness took hold. So this also, of course, impacted our next year of in 2021 for how we were going to approach our research at Pepperwood. Due to the complications of COVID-19, we could not immediately return to this seedling transplant experiment. So what we did instead was pivot, and we decided to go after what might be considered our lower hanging fruit that would still provide us with interesting data and also allow our students to get out into the field and continue this research and these research opportunities. So we returned to our Redwood Canyon site and decided that we would look at the biomass accumulation both above ground and this new era for below ground, at least for our experiment, for those seedlings that had transpired, that had not survived throughout the year. To do this, we removed a certain area around each seedling that we hypothesized would be appropriate for a seedling of that size, then transported these seedlings back to our lab and began the tedious process of sifting out all the soil from the biomass to try and maintain as much of an intact root structure as possible. We then photographed all of these seedlings that we had collected with their georeference data, and then weighed out their biomass for recording a compilation of this data set. Our future will plan to expand on that, and I'll actually talk about that in the next slide. In addition to this removal and accumulation of biomass data, we also expanded the footprint of our 20 by 20 meter plot at Redwood Canyon to a 20 by 50 meter plot, based upon a collaboration that we were able to do with CMPS and their long-term monitoring efforts of the CZU complex fire. This has a twofold benefit. The first is actually that it allows a uniformity of methodological approach between organizations. So we're not losing any sort of data or having issues with replicability based upon how we run our methods. The second benefit comes in from the expansion will allow us to record the effects of a prescribed burn planned for this area later this year or in early 2022. So we can compare what are the effects of fire history on seedling recruitment, if there are any, with our original 20 by 20 meter plot not experiencing a burn, and this extension experiencing a low intensity burn event. So future directions for this are obviously broad. We have taken over the years our results, as well as our collaborative efforts and interests of students in framing new research goals. And I've listed a few here for how we might proceed into the future. Now, I had mentioned that we had plans for compiling a biomass accumulation database for the seedlings at our Redwood Canyon site. But due to the low survivorship and that high mortality rate being experienced at that site, we are very hesitant to remove any of the naturally occurring seeds. And by hesitant, I mean we're just not going to do it. So what we have planned to do instead is the construction and deployment of seed traps to be able to capture the native and the local adult tree population genotypes and plant those in a controlled environment, still at Redwood Canyon, but in a sense creating a sacrificial population of seedlings for the collection of that living biomass data. So we're artificially increasing the population, but in the most natural possible ways, all with the effort to just reduce our impact on the natural system. Another obvious direction that we plan to go with our research is returning, of course, to that Grouse Hill seedling transplant experiment. With that experiment, knowing what we know now, we'll be able to construct controls for both water availability and shade. We potentially can also increase our sample size and the different microclimates that are existing at that site so that we can capture increased variability and see if there might be additional factors that we have not been able to capture with our data sets so far. Another area would be light intensity. I've talked about light intensity, but I haven't really talked about the specifics because our mode of gathering light intensity really only gives us a percentage of light exposure, but nothing to do with the light intensity itself. And Dr. Geary has worked previously, pre-COVID, that is, with student groups at West Valley College to try and develop small modular light sensors that would be able to record data of light intensity inside these plots. And hopefully we'll be able to return to that or find another method for gathering that light intensity as to whether it's the intensity, the amount of light that is received over time. Good questions that we still are not able to answer yet. And finally, and this is a particular interest for me with my graduate work, is expanding the search for physiological and functional traits. Look at the leaf, vascular, root anatomy, and see what the comparisons are between those seedlings that have survived. And also, what Michelle had mentioned, on a healthy redwood population, what kind of stresses are they under that might then impart some detriment or some benefit, perhaps, to new seedlings being produced. So this is just a brief list, but obviously there's almost an infinite number of ways that we can go. And it's very fascinating and very exciting for all the ways that we could actually pursue our research at Pepperwood. And so that brings me towards the end of my talk, but I did also want to take some time to make some acknowledgements. And of course, a big heartfelt thank you to all the Pepperwood Preserve management and staff there. You have supported us and been very generous with us in terms of what we can accomplish with our research there. And we are very humbled and very grateful to you for that support. Michelle talked about this as well, but I really want to say thank you to all the student volunteers that we had, because without them, this project just simply would not have happened. They dedicated their energy and their time for our equipment deployment and our data collection. And it's that unique opportunity that allows them to get in the field while also providing us with a benefit. This mutualistic system is so unique, and it's a pleasure to be able to work with these students. And then, of course, a thank you to all of our donors from experiment.com that helped fund our research and allowed us to purchase new equipment that we use to expand and enhance the questions that we have had at Pepperwood and with our greater research project. And with that, Dr. Geary and I would be very happy to take any of your questions.
To start out, this one I think is probably one that can be pretty quickly answered. There's a definition question. Does redwood envelope, Hugh, I think this is something you were referring to, does redwood envelope mean within the drip line of existing redwoods?
Yeah, that is a great question, and thank you for that. This is a definitional variation depending on who you're asking. For me, what I was using redwood envelope for was the existing footprint of the redwoods as they are at our Grouse Hill and our Redwood Canyon site. So not just necessarily the drip line, but in thinking of what are the outer boundaries of the redwood forest that is there, basically the footprint. And that was what I was using interchangeably with envelope. Perhaps footprint would have been a better way to think about that. But yes, it is, however, still affected by that drip line. And so inside this, under the traditional, the pre-existing forest structure, this whole area that I would say outlined as the footprint did receive the drip line. It was receiving that, but especially at our Grouse Hill site, it no longer receives that. And so the footprint might be a better way to think about those high severity sites.
That makes sense. And I will also add on, for anyone in the room who might not know what drip line is, that would be where moisture that is in the trees in the canopy above drips down onto the soil below, correct? So
Correct.
at
Yes.
Grouse Hill, that doesn't exist because there is no longer a canopy above. Yeah. All right. So we've got another question. This came up during your presentation, Michelle. It surprises me that the range is reducing in the north also, not just migrating northward. Could you elaborate on that? Could you expound on that a bit? Oh, you're muted, Michelle. Yeah.
Well, I would be happy to do that. I'm actually going to share my screen again so that I can go to the map that was being asked about. And so let's see here, move this around a little. So what's being asked about is, if you can you see my arrow? Okay. So why, what's not happy about those locations? What's going on there? This is in Northern California. And what you cannot see on this map, and even if it was larger, you would not be able to see on this version of the map because it's not live. These locations here, those two little red boxes, are both areas where it's higher elevation. So when they were creating their model, and with modeling like this, you're saying, okay, what is this place going to be like according to the climate models, and scaling it down to that particular location? One of the factors they put in is the elevation. And the assumption is that it's going to be cooler at higher elevations. And so what you're really seeing is kind of an elevational effect. There are also streams running through those areas. This is, again, relatively coarse scale. So I would assume that there would be little tiny spots in there that would be next to rivers within watersheds. But these areas are lower elevation. And so basically what the data analysis is saying is these areas are going to get hotter. These areas are going to get drier. Again, not taking the effect of fog into effect, but that's an elevational effect. So it's not so much why are these areas losing, it's why are these areas not losing? And according to their model, they're not losing because of that elevational effect. I hope that makes sense.
Someone said they were interested in propagating redwoods on their own property. And I wonder if either of you have any advice for someone who's trying to pursue that.
So
Michelle, did you want to go start, or do you want me to start?
Why don't I start and then you can jump in? So
That's great.
So if you were in Sonoma County, and I'm assuming that it's likely that you are, but say Sonoma County, you would want to think about whether or not are you in Eastern Sonoma County or if you're in Western Sonoma County. If you're in Western Sonoma County, you could be doing redwood seedlings in areas that are not quite so shady, because you're going to be getting more fog and it's going to be cooler because of the coastal influence. If you're in eastern Sonoma County, I would look for shadier spots, not full shade because they won't like that, but look, in those, have them be in places where they're going to be more in the shade, because otherwise you're going to, the combination of high light and low humidity and potentially higher summer temperatures could be an issue. So a lot depends on how foggy your location is, and sort of where you are in the county. If you're in Mendocino County, so you're going further north, you'd be kind of like being in eastern Sonoma, sort of like being in western Sonoma County. If you're in Napa County, you're going to definitely need to do supplemental watering to get them started, and you want to be thinking about sort of where they are in the landscape. So look at your landscape, look at your property, think about relative to where you are. Do they need more shade and moisture, or do they need less shade and moisture? I hope that kind of makes sense. Hugh, you want to build on that?
Yeah, absolutely. Yes, those are very important factors. And I think I covered this a little bit in terms of our wild populations here at Pepperwood. But of course, understanding that those are in a post-fire site. So that's another factor you'll want to think about, is what is the existing structure that you have and you're planting. The other thing to consider is, what is the age? Are you planting these from seed, or are you allowing these to mature past 1 year or at some other height level or age level and then transplanting these out? Because that will also have an impact. In terms of longevity, it seems that the initial year is incredibly important. The second year is very good. And by the third year, it seems like they have a pretty strong attachment to the soil and are robust enough to be able to resist these low humidity events that we've seen. We currently have in our Redwood Canyon site, which is shaded, a 3-year-old seedling that is very robust and doing very well. But it's not uncommon to have this extremely high mortality event for seedlings. So it's going to have to be considered as to where you're starting from. And then that low humidity, I think, actually is a big factor. So watering is part of this, but if you have too much light or too high of a temperatures, and then that's affecting the relative humidity that they're exposed to, you might consider having something of a shade structure input up, something that might help reduce the amount of heat and then solar radiation on the soil, which is just going to lead to additional stresses on this environment. Sonoma County is one of those drier habitats, and it does shift on these microclimate. This is more the regional microclimate idea of Western versus Eastern Sonoma. But those would be the biggest factors I can consider right now.
Yeah, and that shade structure might be necessary only for the first couple of years while they're getting established. And at that point, you could reduce the amount of shade or potentially take it away entirely.
Correct. After those first 3 years, I would imagine that they should be robust enough to be able to survive on their own in that location.
Great. Okay, we've got a couple more questions that have come in. So since coast redwoods rely on genetic replication, so they send those little runners out, more than seedlings, is the interest in seedling growth mostly to provide genetic diversity?
So I'm gonna jump on that one. So the question of seedlings is interesting with coast redwoods because they are so incredibly long-lived and because they have that potential for resprouting. I don't know that I would say that they rely on genetic replication more than seedlings, but in the short term, that's true. So if you want populations that are healthy across thousands of years, you need to be thinking about, are you getting functional seedlings? Is it the phrase that one of my actually I think this was a phrase from Jen, is it if it's not reproducing by seed at all, it is in a sense a sterile population. It's like a museum piece kind of. So it's a genetic diversity, but it's also, as part of that genetic diversity, thinking about the long-term health of that population and how they're going to be undergoing or not undergoing natural selection. And
then,
yeah.
Oh, I'm sorry, just going off of that too. So the focus so far has been seedlings, absolutely. But I briefly mentioned in our future directions that, in fact, we do want to start looking at some of the adult populations there as well, in terms of what stresses they're under and what kind of seeds they might be producing from that. So there is a genetic diversity. That's why we are focused, absolutely, as Michelle captured perfectly, was this genetic necessity to reproduce. But it's not undue. You're absolutely right that in these more drought-prone sites, that it may be this sort of asexual reproduction or propagation, this kind of extension of these little runners. So we want to follow that as well as the seedlings going forward.
Okay, we've got a couple more questions I'm going to pose to you all, one of which I see Patrice has a hand raised. So Patrice, I'll call on you in just a moment. And then I wanted to present to you, Bruce says the maps project a grim future, perhaps a brighter future for redwoods in New Zealand or Oregon?
So, so again, the maps are based on a certain modeling approach. I'm going to state as a hypothesis, but really it's more than a hypothesis, that if we can maintain healthy redwood forests where we currently have them, those maps will not apply because you'll be able to see survival and seedling production because of the effect of the existing redwood forest. But yes, this is something that is being talked about, is should we be creating other populations? There's a term, assisted migration. Should we be looking at other places and saying, here for other species as well, not just for redwoods, and saying in these locations, they will survive. We will help them get there. We will put them in those locations. How do we do restoration in a world that's different than it was before? Should we be restoring what was there? Or should we be restoring what we think is going to survive? So that grim future, I think, can be forestalled, but creating new populations is something that I think we should also be looking at.
Yeah, that's such an interesting question of like, what should we be planting here, looking at these projections? And on that note, this is veering away from redwoods, but as you mentioned, the idea of like other species like oaks. Someone in the group here is interested in oak tree migration in general, particularly in the Diablo range. And I wonder if either of you have any resources that we could point them towards, or any thoughts on that topic.
There was a paper that came out, and I want to say probably 6 years ago or so. And Michelle Halber, who I think may still be in the group, may know about this as well, that was looking at, for the different dominant tree species, more or less in the Bay Area, what were the expectations? And if you wanted to email me, I could try to track down that reference. It breaks it down not just by, redwoods versus, say, knobcone pines versus oaks. It breaks it down by oak species. So different oak species are projected to have different changes in their ranges in the Bay Area over time. So that there is that work out there. I want to say that blue oak is going to spread a lot, is sort of like my distant recollection. But it's been a number of years since I've looked at that paper. So
on that note, oh, sorry. But on that note, I also would say that I've talked to some researchers at UC Santa Cruz who have looked at oaks. And not only is the migration of these species important, but also their ability to hybridize seems to be very important too, because a lot of the oaks tend to hybridize, and that can be, it's a unique thing that oaks are able to do, and is perhaps isolate, or not isolating, but insulating them to a certain extent against this extreme climate change in terms of not species preservation, but this new hybridization to survive. In addition, and more closely related to pepperwood, my future research is actually more focused not only on redwood populations, but also the mixed evergreen forest and chaparral environments that are associated with pepperwood. And so I will not be directly looking at oak species, but they will be incorporated as they are at pepperwood inside my future studies there. So hopefully there will be more answers, at least at that local, very regional scale, in terms of how that migration is occurring.
So just real quick, the paper that Michelle, the other Michelle posted in the chat, it is one of the papers that I was using. And I guess it was, just about 5 years ago. So that definitely breaks it down in terms of in species. There was also, I just closed that. Hold on a second. There was also a paper that I know Patrick McIntyre was on that was looking at some of those same things, but I would have to, like I said, I would have to go looking for that one.
Yeah, thanks, Michelle. And yeah, so Michelle Hauber, for folks who aren't looking at the chat, the paper that she found the title is California Forests Show Early Indications of Both Range Shifts and Local Persistence Under Climate Change. All right, so Patrice, you've been holding your hand for a while. I'm gonna click to allow you to talk so you can ask your question. If you're still there. Let's see. Oh yeah.
Okay, can you hear me now?
Yes.
Okay, great, and thank you. And it wasn't hard to hold my hand up since I only had to push a button. So my question is a personal one. We have 5 mature redwood trees on our property. We have 2 Chinese redwoods. I believe they're called Dawn redwoods also. And we're in West County in Sebastopol, Sonoma County here. So we're very, very concerned about the change we're seeing in the trees due to the drought. And every year what we do is each tree kind of has its own area around it. So they're not all crowded together by any means, but they're all kind of like sisters. They're close enough that I know that they network with one another. So what we're seeing is that we're seeing a lot of dead needles randomly in the trees. There's still green, plenty of green in them. And when I feel the needles, the green ones don't feel dry. But we're just very concerned about, even though we're getting an amazing amount of moisture in our mornings here in West County, that the drought has gone on for too long and there's been this cumulative effect. So what we do every year is we mulch to try to keep the moisture in for that's in the ground that they can take more advantage of that. So what I've been doing is I've been coming in and doing these 2-hour sprinkler waterings and trying to cover as much of that drip line area as I can. And then it felt like they weren't really getting the benefit. So I dug down under this couple feet of mulch and realized that the mulch was absorbing all my water and it actually wasn't reaching the redwoods. So I did kind of a bold thing. I pulled all the mulch back. And then at night I do this and then do a 2-hour watering, and then I covered it all back up again to try and contain the moisture. So of course I'm a little concerned, did I damage that very delicate area that's right on the surface where, the little mushroom connection is happening and all of that. So I tried to stay, I tried to not dig into the surface itself of the soil. But where I'm at now is, what's my next move? Because it's too labor intensive to keep doing that. I'm worried I'm harming the trees. And we did install a drip line around 2 of the trees recently. And it's irrigation, And so we can actually monitor how much how many gallons are being delivered and all of that. So I wanted to just find out, have I blown it by pulling back the mulch and attempting to water that way and covering it? And then if we do go forward with the drip line around all of the trees, can you recommend how many gallons should we give a mature tree? It's just one big ring of drip line going around it, and I'm actually not able to get all the way out to where the drip line coming off of the tree would be, because cars drive past those areas. So so can you tell me how many gallons and how frequently?
So I'm going to say two things, both of them relatively quickly. I don't think you caused any damage by doing what you were doing. I mean, the redwoods tend to have relatively shallow roots. And so by removing the mulch and getting the water to where the roots are, I think that was a clever move. Because, but realize we are in, we're so sick of this word, we are in unprecedented times. We don't know what we need to do with these redwoods that are in weird locations. I have a redwood in my backyard in San Jose, which is a really freaking stupid place for a redwood tree to be. It's been here for just about 60 years. And I'm doing similar things. I'm watering a couple times this summer. I have no idea how much to give it either. One thing you might try is talking to people at botanical gardens in Southern California, because there are a couple locations of these botanical gardens where they've got redwoods in places that are way outside of the range, and finding out how are they addressing exactly the same problem. I think there's actually a botanical garden, I want to say in LA itself, but I don't know that because I did not go there. Jen went there, we were talking about it. And they're watering to keep them alive, but I don't know how much they're watering. So I'm sorry that I can't give you good amounts, but I don't think anybody knows right now. So go to the fringes.
Okay.
Go to the dry places.
Okay, I appreciate that. Thank you. It's just
Yeah,
agonizing. It's agonizing to see them suffer.
Yes.
It is agonizing to see them suffer. The biggest problem, it too, seems to be the fog events. Even though you're having a lot of fog in the mornings, you're also, it sounds like, adjacent to a road. And that heat that can develop off of pavement can really severely deplete the foliar moisture content. And so you might in some ways be fighting a really difficult battle there with that. And so like Dr. Geary said, really talk to those fringe areas and see what they're doing to address that.
Okay, thank you.
Great. So I see we are just about at time, and I want to, make sure folks can get on with their days. So we'll wrap things up here, and I want to thank you both, Michelle and Hugh, for all of that excellent information, and thanks to all of you here in the room with us today, and for your questions and for learning with us.